Hi, everybody. Thank you for joining me today. Today, I'm going to be doing a short introduction to neurotransmitter testing and talk to you about the importance of monitoring 9 common neurotransmitters via urinary testing. And I find this to be really beneficial when I'm managing the symptoms of chronic disease. So some things we'll cover today. We'll talk about some of the general treatment concepts for optimizing neurotransmitter levels, discuss the typical clinical presentations of individual neurotransmitter deficiencies and elevations, understand the biochemistry of neurotransmitter secretion so that we can support these processes to balance neurotransmitter levels, and then we'll learn how to incorporate neurotransmitter evaluation and treatment into your practice utilizing the NeuroBasic profile. So that we're all on the same page, I do like to start with a simple definition. What are neurotransmitters? Well, we know that they're chemical messengers that regulate emotional processes and physical processes, things like movement, the stress response, pain, and cognition. They function within the central nervous system, but also in the periphery to facilitate communication between the brain and the body's glands, organs, and muscles. Neurotransmitter testing can lead to successful treatment interventions for mental health symptoms. I think we're probably all familiar with this, things like attention deficits, cognitive impairments, mood concerns like anxiety, depression, addictions, compulsions, that sort of thing. But also more symptoms we might think of as physical, like chronic pain or headaches or IBS or weight issues, fatigues, and that sort of thing. Now, I'll say that in my own practice, I feel like everyone suffers from at least one of the following 4 symptoms that I see are super prevalent. Issues with mood, so either anxiety or depression, with energy, not having enough of it, they're super fatigued. People aren't sleeping, so insomnia is really prevalent, and also cognition and attention problems. Antidepressants are often the only intervention offered for anxiety and depression when someone comes in to a doc to seek treatment. You can see here a survey between the years 2015 and 2018 found that antidepressant use increased with age, and it was twice as common among females as males. Despite these drugs being commonly prescribed, a study in 2008 found that paroxetine, fluoxetine, and venlafaxine worked no better than placebo. A meta-analysis in 2011 reported that the relapse rate among depressed patients given antidepressants was up to 57%, while the relapse rate for those given placebo was only 25%. So I think a sensible approach to these common conditions, depression, anxiety, and otherwise, would be to see what their specific neurotransmitter imbalance is before deciding on treatment, because it is important to know what you're treating. Neuroendocrine imbalances often underlie patient symptoms. So when we, as functional medicine providers, are trying to decide where to begin, I think neurotransmitter secretion is an important consideration. Hormones, in terms of sex hormones, super important, adrenal hormones as well. So today I'm going to focus on neurotransmitters, but because neurotransmitter balance and adrenal health are very intertwined, I will be briefly discussing the HPA axis. So let me start this lecture by addressing what is often the elephant in the room, and that is the validity of urinary neurotransmitter testing. I screen for neurotransmitter imbalance with urinary testing. This has been commercially available in the United States for over 20 years, but it is still a new tool to a lot of practitioners. And a common question or criticism of this testing is that it's not valid because it does not solely reflect brain levels of neurotransmitters. And in fact, this is true. Urinary testing is not able to only test what is being secreted in the brain. However, I feel that looking only at brain levels would limit your clinical approach. Neurotransmitters are secreted all throughout the body in neurons of both the central and peripheral nervous systems, and neurotransmitter receptors are found in all tissues, not just nervous tissues. So because neurotransmitters exist in the CNS and the periphery, and we know that the brain and the body are integrated, I feel that neurotransmitter testing provides a good general guide to treatment, and it does correlate strongly with symptoms. And there's some resources here if you'd like to dive in and read more about the validity of testing. So now, since treatment concepts are probably why you decided to attend this lecture, I do want to give an overview of these while we're all fresh. And then after we've done this general overview, I'm going to speak about how to address specific neurotransmitter imbalances. So after evaluating neurotransmitter levels, we can target the imbalances with the specific precursors and nutrients that are needed. And usually this involves amino acids, cofactors, and botanical therapies, specifically nervine and adaptogenic herbs. Amino acid precursors can support low or suboptimal neurotransmitter levels. Generally, I recommend waiting until you've seen the neurotransmitter report before starting your patient on any of these. Generally, these are best absorbed on an empty stomach. So I think of that as at least 30 minutes before or 2 hours after eating. Most peptides and amino acids can freely cross the blood-brain barrier, but whole neurotransmitters sometimes will not cross. So you can see here the ones with an asterisk are both amino acids and neurotransmitters. Generally, these will cross the blood-brain barrier. Sometimes they struggle, but there's certainly still treatment considerations. Adaptogens optimize HPA axis function, but they also contribute to neurotransmitter balance. So of course, adaptogens are herbs and even mushrooms that support the body in resisting and adapting to stress. Officially, to be called an adaptogen, these must enhance physiology without any adverse effects. Adaptogens will promote optimal hormone and catecholamine levels, whether your patients have started with low or high levels. And we know that adaptogens promote balance to the entire adrenal gland, both the cortex, which produces cortisol, and the medulla, which produces some of the neurotransmitters. And then nervine herbs are also a consideration. These herbs help to restore balance in the nervous system to help patients cope with stress and anxiety and insomnia and even mild depression. Now, Mucuna pruriens is a great herb to have in your tool chest. It's an Ayurvedic herb that's been used to treat Parkinson's in India. It contains L-dopa or levodopa, which is that precursor to dopamine, so it can support low catecholamine levels. And St. John's wort is also an herb I want to highlight because it's been shown in studies to provide equivalent relief of mild to moderate depression when compared to SSRIs. So this can certainly be considered a treatment option for mild depression. I do want to remind you to use it with caution because St. John's wort can cause drug interactions because it's metabolized via some common cytochrome pathways. L-theanine, this is a really helpful amino acid because it has many mechanisms of action. It's found almost exclusively in the tea plant. It can be used to treat both high and low neurotransmitters, and it's recommended often. So some of its functions include action as a GABA agonist, so it has neuroinhibitory and parasympathetic effects. It can counterbalance the excitability of glutamate and can modulate serotonin, GABA, and catecholamine levels. Theanine has a pretty broad therapeutic window, so you could consider between 100 and 500 milligrams twice a day. You do want to think about giving it twice a day because it has a shorter half-life. Some people like to treat with food, and so you could consider giving actual green tea to give your patient theanine. But I do want to just point out that a cup of green tea only contains between about 10 and 50 milligrams of L-theanine, depending on the grade of tea. So your patients would have to drink a lot of green tea to get to the dosage that have typically been found to be beneficial. Now, when it comes to mindfulness and meditation, there have been a number of studies that evaluate the effect of regular meditation on neurotransmitter levels. They found higher serotonin levels and lower norepinephrine levels when people who meditate are compared to those who do not. Meditation can also increase GABA, and higher melatonin levels are seen the night following meditation. Now, let me speak quickly about cofactors. So these are the substances that are essential for an enzyme's activities. Clinically, they become important when conversion is slow. So let's look here at the conversion between norepinephrine and epinephrine. If norepinephrine were elevated and epinephrine were low, we would have to consider that the PNMT enzyme might be a little bit sluggish. And if that were the case, we would consider giving the cofactors that help to fuel the activity. In this case, we're looking at SAMe, or essentially methylation, as well as cortisol. So looking at HPA axis function might be of benefit here. Now, these are the biochemical pathways of the neurotransmitters that are tested in the NeuroBasic profile. You can see here, if we look through the lens of cofactors, the cofactors are listed in orange. These are often minerals or vitamins, especially B vitamins. You can see the neurotransmitters that we test are purple, and the enzymes are here in blue. Now, methylation is actually really important consideration with neurotransmitter imbalance. Methyl tetrahydrofolate reductase is the name of both the gene and the enzyme that play an essential role in processing folate. And methylation affects a diverse variety of metabolic processes. And so when those are interrupted, you may disrupt neurotransmitter creation. People who have faulty MTHFR gene mutations make faulty MTHFR enzymes, and so they can experience interruptions in methylation pathways. And essentially, we commonly see low to low range levels of serotonin, dopamine, norepinephrine, epinephrine, and glycine. Now, epinephrine is italicsed here because even in patients who have an established defect, I have often seen epinephrine elevated when the others are not. And I think that this is because of the importance that epinephrine has in the stress response. People who are poor methylators are not typically unable to methylate, it's just compromised. And so sometimes all of the methylation seems to be funneled into epinephrine because of its importance in that stress response. You can test for MTHFR variants, or you can choose to treat empirically, and some options for treatment are listed here. Now, vitamin D is also an important cofactor that activates gene expression of both the tryptophan hydroxylase and the tyrosine hydroxylase enzymes, and these are the rate-limiting steps for the production of serotonin and dopamine. And fish oil is very important. It's not a cofactor, but it does benefit the nervous system. So there are two major components of fish oil, EPA, which is very anti-inflammatory, and DHA, which provides fluidity to cell membranes, and so it's vital to brain and nervous system function. Inflammation often underlies chronic health issues, and it's been associated with depression specifically, so fish oil can have antidepressant effects. Probiotics, or the microbiome, also need to be considered. Studies in the last decade have demonstrated a bidirectional relationship between the gut microbiome and brain function. Researchers are calling this the microbiotic gut-brain axis. And we know that when the gut microbial composition is altered, we can see changes in mood, pain, and cognition. And dysbiosis has also been implicated in things like depression, anxiety, IBS, and autism. And there are a lot of studies here if you'd like to dive further into this gut microbiome brain connection. This was an interesting little study because they looked at how probiotics can attenuate anxiety and depressive-like behaviors in experimental animal and human models. Evidence suggests that oral probiotics may have beneficial consequences on mood and psychological distress. And the authors proposed three potential mechanisms: competitive exclusion of deleterious gut pathogens, decreases in pro-inflammatory cytokines, and communication with the CNS via vagal sensory fibers. So all of these proposed mechanisms can affect neurotransmitter secretion. Not every depressed person is inflamed, but inflammation is a common avenue to depression that should be considered when trying to determine the root cause of neurotransmitter imbalances. This article in Psychiatric Times from 2018 lists some helpful considerations when treating mental health, including assessing for a common cause, which is inflammation. And you can see that they're recommending implementing some of the lifestyle interventions that we functional medicine docs always recommend, and reducing inflammation is there at the top of the list. So this is the NeuroBasic profile. If you're interested in assessing neurotransmitter imbalance, starting with the NeuroBasic profile is a great option. This is an AM spot urine collection. In the NeuroBasic profile, we test serotonin, dopamine, norepinephrine, epinephrine, GABA, glutamate, glycine, histamine, and phenethylamine, which we generally call PEA for short. And you can see here their results are given to you in a numerical value, but also plotted out on this histogram. Blue is frankly low, red is frankly high, and yellow, green to yellow is within the reference interval. The yellow zones representing sort of those cautionary zones when levels are trending high and trending low. So now let's get into the details so we can adjust treatment plans based on a specific patient's results. And we'll start with serotonin. So biochemically, serotonin is derived from the amino acid tryptophan. It's actually, even though we know it's very important for mood, it's primarily found in our gastrointestinal tract. Up to 90% of the serotonin is secreted in the gut, and the remaining 10% or so in the platelets and the CNS. Now, some but not all symptoms of imbalanced serotonin are listed here. Things like body temperature changes, so hot flashes and night sweats are actually associated not just with hormone levels, but with serotonin levels. Pain, depression, suicidal ideation, racing thoughts and obsessions. As we move through the types of symptoms you might expect to see with various neurotransmitter imbalances, I want you to note how often the same symptoms will appear. For instance, you might see mood concerns associated with many neurotransmitters, cognitive concerns associated with many different neurotransmitters. And this is one reason why I find testing to be so essential. It really helps us pinpoint what in particular is contributing to a patient's symptom pictures. Now, with serotonin, appetite change is a really common finding when serotonin is imbalanced. So you might see a lack of interest in eating, or more commonly cravings, and specifically for sweets and simple carbs. This seems to be an attempt at self-medication to raise serotonin levels. What we know is that often neurotransmitter imbalances will drive behavior. So as we move through this lecture, I'll have a treatment slide for every neurotransmitter we discuss. You can see treatment ideas for treating low levels on the left and high levels on the right. So these are here for your reference. So let's move on to dopamine. People with extroverted personality types tend to show higher levels of dopamine activity than people who are introverted. This neurotransmitter stimulates the reward and pleasure centers in the brain, while also affecting movement and behavior, allowing us to move toward an unexpected reward. And this reward response is enhanced, especially if the reward received is greater than expected. So think back to when you were a child and you put a quarter into the gumball machine and you turned the crank and a shiny red gumball dropped into your hand. That was fun, but not necessarily super exciting because you expected to get that gumball. But if you ever had the experience of putting the quarter in and turning the crank and having two shiny red gumballs unexpectedly drop into your palm, and the excitement that you felt, that unexpected reward and that excited feeling, that was dopamine. So dopamine is a catecholamine that's derived from the amino acid precursor tyrosine. It then converts to the other catecholamines, norepinephrine and epinephrine. So this is how people with excess dopamine may see the world. Things that once seemed safe and comfortable no longer do. Symptoms include increased worry, paranoia, ADHD, a decreased ability to interact socially, and a desire for instant gratification. Conversely, dopamine deficiency is associated with things like Parkinson's disease, mood swings, depression, anxiety, and a loss of motivation, which can include sex drive. With low dopamine, a person may be more susceptible to developing behaviors that result in a temporary increase in dopamine. And generally, these are risky things, potentially dangerous things like drug abuse, alcohol abuse, addiction to food, addiction to gambling, addiction to sex, etc. And just like with eating carbs might result in a little serotonin boost that makes people feel better, when it comes to dopamine, engaging in these risky behaviors can result in a little bit of dopamine boost, which makes people feel better. So these are sort of self-medicating behaviors. Dopamine is sometimes called the motivation molecule. Lab mice that are dopamine deficient are so apathetic they will literally starve themselves to death, even when food is readily available. That's how important dopamine is to motivation. And here are some treatment options if dopamine is low or high. So moving downstream from dopamine, we have norepinephrine and epinephrine. And of course, these regulate the fight or flight response. So we think about attention and arousal, regulating heart rate and blood pressure, and releasing glucose from energy stores. So together, norepinephrine and epinephrine coordinate that fight or flight response. They're released from storage vessels in the adrenal medulla in response to stress and increased degradation of glycogen and triacylglycerol, as well as increased blood pressure and cardiac output. So they help us get ourselves away from the dangerous situation. So what do these people have in common? They are adrenaline junkies. So adrenaline is the same thing as epinephrine. It's just a different verbiage. And outside of the United States, epinephrine is often referred to as adrenaline in the medical literature. When catecholamine levels are low, some people engage in self-medicating behaviors. And when we think about these that are involved in the stress response, epinephrine and norepinephrine, what these people will do is activities that self-induce a fight or flight response. So I do want to note it's perfectly fine for any of us to do exciting things with our time. But what we're talking about here is the people who don't really feel alive unless they're engaging in these risky activities. At this point, we might consider intervening. Because catecholamines are excitatory, low levels of norepinephrine and epinephrine can lead to things like poor attention and memory, decreased concentration, reduced socialization, loss of alertness, brain fog, etc. And conversely, elevated levels can lead to physical symptoms like palpitations, arrhythmia, restlessness, hypertension, mood symptoms like anxiety, lack of focus, essentially being overstimulated. And then support for norepinephrine and epinephrine are here for you. Now, the norepinephrine to epinephrine ratio is something that's calculated for you as part of the neurobasic profile. So this examines the conversion between norepinephrine and epinephrine. This is one way to assess how stress affects neurotransmission, because cortisol is a cofactor for the conversion between norepinephrine and epinephrine. So if we have a lot of norepinephrine and not a lot of epinephrine, we'll have an elevated ratio. And this indicates that you may want to look into cortisol secretion or HPA axis functionality. So when we think about the stress response, I think about 3 phases that are important to identify. Now, these are not black and white, but this is how I often think of it and explain it to my patients. Because patients can often present with the same symptoms, right? They can be fatigued, depressed, anxious, have trouble focusing and trouble sleeping. All of these symptoms can present at different phases of the stress response because everyone fatigues at different rates. So I think it's important to test, because depending on the stage of the stress response, treatments can vary quite a bit. So in this early stage of the stress response, or phase 1, typically what we see is elevations in cortisol as well as epinephrine and norepinephrine. So in this initial stage, the body is essentially very efficient at responding to stress. But the sympathetic nervous system is not designed to stay in high stress mode continuously for months and months. Some people might call this adrenal fatigue, but the adrenal gland isn't really fatiguing. Rather, what we're talking about is that sympathetic response and the HPA axis adapting to the constant onslaught of stressful stimuli. And so at this point, we may see cortisol levels start to slip, and some of the catecholamines may start to become depleted. Now, I will say epinephrine tends to be the last of these markers to decline because it's so important in the stress response. And then patients can move into late stage HPA axis dysfunction, where they are actually depleted and exhausted. So here we see very low cortisol secretion as well as low catecholamine secretion. Now, of course, like I said, there are no hard and fast rules governing these changes, but generally these are the stages that reflect what I have observed in patients who are dealing with chronic stress. All right, back to the neurotransmitters. Moving on to glutamate, which is the most abundant excitatory neurotransmitter in the body. It's molecularly similar to GABA, which you can see here, but the two have opposing neurological effects. So that's always been very interesting to me. Since glutamate is excitatory, pathologies correlated to high levels include anxiety, insomnia, ADHD, and poor concentration, whereas low glutamate levels are associated with depression and brain fog, addictions, and learning difficulties. MSG and glutamic acids in foods can contribute to glutamate elevations. And so if you find elevated glutamate on your patient's test results, you may want to talk to them about their diets. And here are some ideas for supporting low and high glutamate. Now, moving on to GABA, whereas glutamate was the primary excitatory neurotransmitter, GABA, its first cousin, so to speak, is the major inhibitory neurotransmitter. It's relaxing and calming. Now, it is synthesized from glutamate via the GAD enzyme with the help of pyridoxal 5-phosphate, which is activated B6. The GABA-A receptor is the prominent receptor, and it's the same receptor that's utilized by the benzodiazepine drugs. And so stimulating these receptors can help to treat things like anxiety, can act as sedatives or muscle relaxants, or even helpful in treating things like seizure disorders. Remember, because GABA is inhibitory, if it's low, we might see excitatory symptoms like panic, anxiety, or depression. And if GABA is elevated, we will see dampening symptoms, right? Drowsiness, difficulty concentrating, diminished memory, decreased cognitive processing. Now, one little pearl here, GABA levels can become elevated as a compensatory mechanism when excitatory neurotransmitters are high. So even in the absence of supplementation, if there's a lot of excitatory activity, maybe glutamate or the catecholamines, the body may raise GABA levels as an attempt to maintain homeostasis. So elevated GABA is not necessarily a bad finding. And here are some ideas for addressing imbalanced GABA. Now, moving on to histamine. Histamine is an excitatory neurotransmitter that's involved in the sleep-wake cycle and the inflammatory response. It plays a dual role in the body as both a neurotransmitter and an immunomodulator that increases metabolism, promotes wakefulness, and suppresses appetite. And its precursor is the essential amino acid histidine. Now, when we think about the neurotransmitter uses of histamine, I typically think of it in terms of its ability to impact insomnia because it increases wakefulness and prevents sleep. If histamine is low, we can see drowsiness, changes in digestion and appetite control, and issues with learning, memory, and mood. And then if high, like I said, increased wakefulness and insomnia is really the slam dunk here. But we could see symptoms that are typically happening outside of the CNS, things like allergy-like symptoms and then gastrointestinal concerns. And ideas for supporting low and high are here. So let's move on to glycine, which is inhibitory, and it helps regulate the body's temperature during sleep. Elevated levels are typically associated with poor cognition, whereas low levels may contribute to poor sleep and memory issues. And some treatment considerations are here. Now, interestingly here, bone broth contains a lot of glycine. And so if your patients are low in glycine, it's a great option as a therapy. But also if they have high glycine, you may want to quiz your patients and see if they're getting things like bone broth or collagen supplements in their diets, because those do contain a lot of glycine naturally. Now, moving on to the last neurotransmitter that we test in this NeuroBasic profile is phenethylamine, or PEA for short. PEA is stimulating and excitatory. So if there are low levels, we often see ADHD, depression. It's even been associated with things like autism and Parkinson's. And high levels, because of its excitatory nature, can be associated with anxiety, nervousness, paranoia, aggressiveness, diminished appetite. Now, one of the main things that I look for with PEA is if it's low, it has been suggested in the literature that it can be considered a biomarker for ADHD. So 3 studies have confirmed that urinary PEA levels were significantly lower in patients with ADHD, and that these levels normalized with treatments, the amphetamine-like treatments for ADHD. So now researchers are suggesting that urinary PEA levels could actually be considered a biomarker for ADHD. So if cognition is a particular concern for your patients, you definitely want to make sure you're taking a look at PEA. It's not always the culprit, but it certainly could be. And here's some ideas for supporting both low and high PEA. And if you want to dive into the references for supplementation, they're here for you. All right, so when looking at the neurotransmitter report, in addition to looking at the individual highs and lows of these markers, you do want to consider the relationship between excitatory and inhibitory balance, because this can help you put symptoms into context and prioritize treatment. So the neurotransmitters that are considered to be excitatory are glutamate, that's the most prominent one, followed by dopamine, norepinephrine, epinephrine, histamine, and PEA. The inhibitory neurotransmitters are GABA and glycine. Now, I have serotonin over here sort of on its own. As a precursor to melatonin, I think it could potentially be considered inhibitory, but I think of it more as sort of a neutral neurotransmitter. It's certainly not excitatory. Okay, so I hope that this overview has provided a roadmap to optimizing neurotransmitter balance in your patients, because so many of my patients present with fatigue, mood concerns, issues with focus and concentration, and sleep concerns. I think that this is a fantastic tool to help me tease out the individual imbalances that can contribute to these common symptoms. The etiology of symptoms like these is really complex, and so it can be different in each case. Assessing neurotransmitter secretion via the neurobasic profile can make a significant difference in understanding and treating the patient who is sitting right in front of you. All right, thank you very much for your attention today.